Good morning, everyone. It is a moment of extreme pleasure to welcome you all on the behalf of Department of Biotechnology, V. Narmad South Gujarat University, Surat. Active involvement with the alumni provides career guidance to the current students in their professional life. These engagements enrich the overall experience of the students and provide them with a competitive advantage in the current global world. Department of Biotechnology has organized a first webinar in the history of our department aiming the interaction of current students with the alumni. It's our holy tradition to worship our goddess Ma Saraswati before any important event. So let's dedicate a prayer to Ma Saraswati. I request the students come forward for the prayer. Come forward.
या कुन्देतु तुषारहारधवला या शुभवस्त्रावृता या वीणावरदण्डमण्डितकरा या श्वेतपद्मासना या ब्रह्माच्युतशङ्करं प्रभुदिभिः देवैः सदा वन्दिता सा मां पातु सरस्वती भगवती निःशेषजाड्यापहा.
Thank you. So now, I would like to invite our coordinator, Sir, Dr. Gaurav Shah, to address the audience by his words of wisdom. Good morning,
Thank
sir.
you. Good morning. Good morning. Thank you very much, Mansi. Dr. Mansi is also alumni of this department, and the talk which we have organized today is related to the applications of CRISPR-Cas9 in the field of crop improvement. The most important thing is that the initiative which we have taken is related to current students and alumni interaction is not only for the benefit of our students, but now with this online platform, this digital platform, the expertise in the field of our students will be benefited to others also. And I really thank Dr. Mansi Mehta for taking this initiative, being the alumni of this department of first batch. And the speaker today, whose introduction will be given by Mansi, is also our alumni from second batch. So I welcome you all on this digital platform, and I thank all of you for being a part of this journey in this department. And as Mansi already told, that this is the first webinar digital platform. In case of any technical glitches, I as an head of this department, I apologize, and I am sorry if at all something happens, something goes wrong. So my best wishes to all our students, as well as those who are going to benefit with this talk related to the crop improvement. Thank you very much.
Thank you, sir, for your words. So let's move forward. I would like to introduce our guest speaker, Ms. Hiral Lakhani. Welcome, Hiral. Welcome, Hiral.
Hello, sir. Hello, Mansi. Hello, everyone.
Okay.
Am I audible to everyone?
From batch 2, that is in 2012, is going to address on a case study for application of CRISPR-Cas9 cpf1 in crop improvement. Currently, she is working on CRISPR-Cas9 genome editing uh, as PMRF Scheme Fellow at National Agri-Food Biotechnology Institute, Nabi, Mohali, and she is also working on CRISPR-Cas9 multiplexing as well as she is working on protoblast isolation and regeneration for transgene-free genome editing in banana. So, Hiral, are you ready for the webinar?
Yes, yes.
Okay, fine, fine. Okay, fine.
Okay,
Let's start our session, and I'm going to share the PPT.
okay. And am I audible to everyone, Mansi?
Yes, yes, yes.
Okay, let's start. First of all, thank you, sir, and Mansi for organizing this and giving me this opportunity to interact with the students. If I give my introduction to the students, then as already Mansi and sir has addressed that myself, Hiral Lakhani, and I am the alumni of batch 2012 from the Department of Biotechnology. I have done my bachelor and masters from the department itself, and after that with the help and during the study and after the study also with the guidance of sir and other faculty, they have always motivating and encouraging us for to give various competitive exam and all the things. So I am grateful to the department uh, for being at the position that I am. So let's not waste time, and let's start this session. So if you guys have any queries or something like that, then you can comment or tell to the Dr. Mansi, and we can address all your queries at the end of the session. So let's start. First is the, as the name suggests, at a case study for application of CRISPR-Cas and cpf1 in crop improvement. So might be, as all of you are master's student, then you must know about the name of CRISPR, and this is actually highlighted recently in such a way, in a very large way, because in the recently, that in the year of 2020, the Nobel Prize in Chemistry was to this CRISPR-Cas9 technique for the, and that to the two ladies, Jennifer Doudna and Emmanuel. That's why this technique is very important, and today also I will let you know that why this technique is so important and why it is widely used in the for the not only crop improvement but for all the different organisms in worldwide. So next, see, before going on to genome editing tool, I can say that as the name suggests, you must know that what is genome editing. So, as the name suggests, that is the changes or we can say addition, deletion, addition, insertion, anything to the DNA sequence of any organism on the earth, whether it is that is ranging from bacteria to the animals. Okay. And first of all, why this thing is needed? So because of the we can say that because uh, see why this genome editing technique is needed, because we in the global situation, we know that especially if I talk about the crop, then we want because of the uh, global population is increasing, and we have very scarcity of food, and apart from that, the type of crop that are present, because of the climate changes, they are affected by pest, they are affected by the climate. So resistance to the environment, resistance to the abiotic stresses, resistance to diseases, for all these things, or we can say improvement of the crop, for all these different purposes, we need genome editing. So even before going into the genome editing tool, इनिशियली और वी कैन से पहले के टाइम पे अगर हम स्टार्ट करें तो इनिशियल द टेक्निक वाज ब्रीडिंग एंड इन फैक्ट ब्रीडिंग इज द नेचुरल टेक्निक even before knowing the science. In nature it was there, like breeding was there, or we can say mutation was there in the environment, but everything was very in a slower process. But after that we came to know about the cross pollination, etc., and we human have started cross breeding, and in that way we have improved the many traits of many different crops. But that is also not sufficient, and in the recent time, as the science grow, as the molecular biology techniques grow, we came through various different techniques, such as I have mentioned over here, such as ZFNS, that is zinc finger nucleases, then talent activator like nucleases, oligonucleotide directed mutagenesis, and the latest one is the CRISPR-Cas9, or we can say CRISPR-Cas. Cas9 is one type of the CRISPR, and there are a lot of disadvantages of this technique. There are certain disadvantages of CRISPR as well, but it has comparatively advantages as compared to other techniques because it do the things in a very precise manner. So let's go detail into the CRISPR. Here is a quite introduction, a very small introduction. That is, use of this technique is used for the site specific nucleases, and it has demonstrated precise genome editing, as I already stated, in animal and plant system. And here also this genome editing or CRISPR especially work in NHEJ, and another is the HDR. I will let you know about what this non-homologous end joining and homology directed repair in the next slide. So, and apart from that, as I told you that you have also come to know about the transgenic plants. We already have a very many. There is a list of different transgenic plants that are present globally. If I give one example, then that is the BT cotton. Okay, you must all have studied about the BT cotton, and BT cotton is also used in the. It is grown in the India, and its production is also giving many benefits to the farmers. Okay, but apart from that, there are also lots of different edible crops are also there, which were modified with the help of other techniques, but such as the bt brinjal. Okay, but bt brinjal is not introduced in the India. Why? Because it contains the transgene. As the name suggests, transgene, that means this gene that has been introduced from something else. Not like if I say that it is not from one brinjal to the one species of brinjal to the another species of brinjal. No, but it is from the different species, like it has been introduced from the bacteria to the brinjal. So this is the thing that is referred as the transgen. And in India and in many of the country globally, transgenic plants are not especially used in the for the eating purpose. Why? Because we don't know that what that introduced foreign DNA in that crop will do when it will enter in the human body. Okay, so that is the mainly reason. And CRISPR is also highlighted or widely used in entire globally because it has the capacity to eliminate this transgene word and doing the precise gene editing. Next, see, this is the origin of CRISPR-Cas system. So if I talk about little bit history of it, then this entire thing or this entire story has been started back in the 2006. Like it was initially, it is, you can say that many of the discoveries are accidental, but we can say this is not an accidental discovery, but this is like, we can say that in science, what the main thing you need is the observation. Okay. So some scientists, they have identified that okay, before going in that, you all must know about the viruses, that is the bacteriophages, right? So bacteriophages, as its name suggests, that they eat the bacteria, right? So while doing or while studying on the viruses, we know that once the bacteriophages enter inside the bacteria, they can kill the bacteria. They will multiply in the number, and once their number of viruses achieve, then the cell burst and all the viruses will be lynched. This might you have studied. Okay, this is the concept behind the bacteriophages, right? But during the study on viruses, what scientists found that after introduction of the virus DNA inside the bacterial cell, there is a type of, or we can say that what bacteria did what scientists identified, that bacteria have adapted an immunity in a certain way that they will not allow this bacteriophages to enter, they will not allow this bacteriophages to multiply or to replicate inside the cell. And that is what here it is referred as a bacterial immunity. Okay, now how scientists find out this? It took a very long period. Like the things has been started in the 2006, and in the 2012, as I told you about the two name of lady, that is Jennifer Doudna and Emmanuel. What they found out, that they found out certain type of CRISPR array in the bacterial genome. Okay, now what this CRISPR array is that? See, this CRISPR. The full form of CRISPR will say many things about it. Like the full form of CRISPR is the clustered regularly interspace short palindromic repeats. Okay. See this, the square box that you will find, that they are the kind of a palindromic repeats. You must know about the word palindrome. Palindrome, that means a type of a double stranded sequence which is similar in the forward and reverse direction, right? So the square are the kind of palindromic repeats, and this diamond shape in between the structure, that is the sequence of viruses that has entered. So here the things will come. Okay, what bacteria will do, bacteria will identify and recognize once they will find that something foreign has been entered, some foreign DNA has entered inside the cell. So what they will do, they will identify one particular segment of the DNA, and it is particularly ranges from 20 to 24 base pair. And what bacteria will do, they will identify and incorporate this sequence with the help of recombination in their own genome, but not at random places, but they will incorporate these things in between in the CRISPR array only. Okay, and next, this is the thing, or we can say that they have adapted the things, they have adapted the immunity, and at the same time or in the next time when they will find out, or the same type of sequence they will find that it has been entered inside the cell, what they will form, they will form the CRISPR RNA form. What will happen over here, here here I can say that this is the DNA sequence of the viruses that has entered, and these are the palindromic repeats. This thing will be transcribed and it will form the CRISPR RNA. Now, there are two main elements are there in the CRISPR system. One is the CRISPR RNA. There is two terminologies are there, CRISPR RNA and tracer RNA. Together here also, and in the future slides also, I will refer it as a single guide RNA. As the name suggests, that is a guide RNA. This is the first part of that. The second part is referred as the Cas9, or we can say right now I will say that Cas because it is a system only. Cas9 is one part of CRISPR-Cas. Okay, so this Cas, it stands for CRISPR associated protein. One function has been done by the CRISPR RNA, and the another function is done with the help of Cas protein. This Cas9 protein is also part of this CRISPR array. Once there is the formation of a single guide RNA, the single guide RNA, it will go and it will search the same type of this guide RNA sequence in the virus genome. And once it will find that yes, there is something, or they will find that homology, then it will bind with it and the Cas9 protein will cut it. So this is the basic technique. And once it will be cut it, there will be no any further virus replication or anything will be happen, and in that way bacteria will save themselves. So we can say this is the system that is adapted by the bacteria to save themselves or to adapt the immunity. We humans have identified this system, and we are going to mimic the same system in other organisms, such as whether it is bacteria, plants and animals. We will mostly refer today is about only the plant, but this is the system that is going to utilize by humans. So I hope that I'm clear about clearing your concepts. Again, this is CRISPR loci. As I have already mentioned about the things, here the things are clearly visible. These are the spacers, and the spacers are the different type of sequences that has been introduced in the bacterial genome, whether it is from virus, whether it is from plasmid or anything. So this CRISPR loci, it can be it can be differ in a, or it can vary in the length. Okay, and the other thing here is the Cas gene. They are also present in the same channel or same locus, we can say. So once there will be formation of the CRISPR RNA guide, that is the guide RNA, there will be also at the same time transcription of this Cas gene, and ultimately translation of the Cas9 protein. So this is how the things work. See here also, as I already mentioned, that a short piece of viral DNA that get integrated into the CRISPR locus as per the spacer, and it ranges from 21 to 72 base pair, but mostly it is ranges from 20 to 24 base pair. And this is a spacer, that means, as I already said, that spacer, that means the genome of viruses. Okay, and CRISPR repeat, that means the palindromic repeats that are present in between the spacers. This repeat, it ranges from 24 to 48 base pair, and it is different in case of different bacteria. And again, if I say that this system was first recognized in the bacteria, that is SP-Cas9 and streptococcus, and that's why this name is also referred as the SP-Cas9 also. And after the SP-Cas9, there is a list of different bacteria in which scientists had found the same type of system, but initially it was the SP-Cas9. So this repeats, as I already said, that they are palindromic and encouraging the secondary hairpin structure. Repeats are separated by the spacer of similar length. And during the RNA transcription, see here it is, we can say that this is a transcript. This is the primary transcript. And after that, this palindromic repeat, it will form this structure in a certain way that it will form a secondary loop structure. Yes, this type of secondary loop structure will be formed. So this is a target sequence, that is a spacer sequence, and this is the palindromic repeat, and together they will form this type of structure, and this cas9 have a perfect homology to fit this particular guide RNA inside them. And once they will identify the target sequence, this cas9 has a cleavage activity, and it will cleave or it will chop the DNA where it is present. So, in summary, I can say that it is consists of two things. One is the CRISPR RNA, one is the single guide RNA, which made up of CRISPR and tracer RNA. This is theoretically. When we are practically performing it, we are not going to design or synthesize two different type of RNAs, but instead of that, we just form a one single guide RNA in which we have both the things like CRISPR RNA and tracer RNA. And the another major part of CRISPR system is the Cas9 protein, that is endonuclease that cleave the DNA. It have a certain type of domains, active domains, that has the capacity to cleave the DNA. Okay, so to create a gene disruption, a single guide RNA is generated to direct the Cas9 nuclease to specific to any of the genomic loci. And Cas9 induced double-stranded break, they are repaired via, this is very important things. See, once here I can say that okay, we have there is a designing of guide RNA, Cas9 protein is there. We have prepared the assembly, and it will find its target and it will cleave it. Okay, but what happened after that? That is the main story comes after the cleaving of this. I can say, or we can say that there is the, see, there is two different type of repair pathways are there. Okay, you must all have studied that whenever there is cutting of DNA is there, or any type of damages happen in the DNA, our cell or our nucleus is very smart. It has all the different type of machineries that how to repair this thing. Okay, and once they will try to repair by themselves. So once there is a cutting or activity of, due to the activity of cas9, there is a cutting or chopping of our target DNA, then two different pathways will be activated. One is the NHEJ and another one is the HDR. Globally, mostly utilized is the NHEJ. The full form of this is the non-homologous end joining, and HDR, that means homology directed repair. NHEJ, it is the iron prone pathway that is commonly present in all the living organism on the world. And what it means that once there is any type of cutting is there, then cell will be activated and it will try to ligate it. Okay, so what it will do, NHEJ, what it will do, it will double stranded break recognition. Once it will recognize that there is something type of break or something has happened to the DNA sequence without any uh, precise thing, then it will first recognize it. Then processing of the non-ligated DNA termini, that is like here it is, there is, we can say that cutting is there. Okay, so it will recognize it and it will try to ligate it, and while trying to ligate it, it will not see that what the type of sequence was already present. No, what it will do, it will just try to repair it. Okay, so in that cases there is certain type of insertion of some nucleotide, some deletion of nucleotide. So this is the thing that what will happen here, that the gene in which we have targeted the things, it will be disturbed. Okay, it can disturb in the sense that you have studied about the central dogma, like DNA, then RNA, and then protein. Okay, so you have also studied that whatever the sequence present in DNA, it will be transcribed and ultimately protein will be formed, right? But what if there is problem in the DNA, or we can say there is a type of deletion or addition in the DNA, no transcript will be formed and no protein will be formed. So what we can say, that the disruption of that gene has been, disruption of the gene function will happen, and there will be no any protein will be there and no functioning will be there. So what we can here say that we are we can say that we are breaking the function of that gene, and ultimately we will find some type of phenotype or any type of misfunctioning of any other things. Okay, so that is the reason that why most of the people, or globally most of the people are trying to study these things. Okay, and another thing is the HDR. As I suggest that HDR, that is the homology directed repair. What will happen over here, that this is quite difficult, and it is complicated also, designing and everything, but yes, some scientists have achieved these things also. In this, what they will do, here there is a single guide RNA and Cas9. In case of HDR mediated repair, there is also one more homology sequence will be there. Now this homology sequence, it it have this sequence which will be compatible to this thing, or I can say for this thing. Like here what will happen that I can say that one gene is responsible for producing certain number of grains in the rice. Okay, but I want that number of grain of rice will increase. So what I will do, I will first identify the gene which is responsible for the number of grains. And what I will do, I will modify that gene, or I will modify its sequence in a certain way that that gene will be overexpressed, or it will more amplified, or in certain way that it will increase the number of grain. So what I will do, I will identify that particular part of gene which is mainly responsible for ultimately grain number. And I will at the same time, I will also synthesize the DNA sequence which I want to incorporate in the DNA. So in that case what will happen, again Cas9 will come and it will do it activity. And here if that cutting place, instead of random insertion of any addition or deletion of any nucleotide, here there will be incorporation of my donor template only, and this donor template sequence I will decide. Okay, so here there will, we can say here, that's why the word is written, that is a precise modification, because this modification I have selected, and now this gene will function in certain way that I want. Okay, I have just given you one example. You can do whatever you want with the help of this thing. So, a distinguishing feature of this CRISPR system compared to other sequence-specific bacterial defense, that this system is comparatively adaptive, as I told you why it is adaptive. Okay, and a specific Cas protein recognize foreign genetic material and integrate their DNA as a new spatial sequence in the CRISPR array, ultimately allowing the bacteria to adapt and subsequently target this foreign element. Okay, again the same thing that I have already mentioned you. Okay. I told you that there is only 20 base pair sequence that bacteria will or guide RNA will identify in the target sequence. So let's go back a little bit. See here, this thing is there. This is the guide RNA, CRISPR RNA, tracer RNA. Okay. I told you that yes, you have to design a guide RNA or bacteria. Remember this 20 base pair sequence. It will try to match the sequence wherever it is possible in the bacteria. Once they will find their homology, they will cut. But here, the main important thing, again, one more important role come, that is of the PAM sequence. What this PAM stands for? Protospacer adjacent motif. This guide RNA, it will go and try to see their homology, but it will bind only at the place where there is the presence of a PAM sequence. And this PAM sequence, it is present downstream of our guide RNA. Like if I say that this is my guide RNA, here it is my 5 prime, this is my 3 prime, then this PAM sequence, it will be over here. This PAM sequence is the sequence which is present in the target sequence only. यहाँ पर हमने और कुछ अलग नहीं किया है. Okay, so here there is the PAM sequence. Guide RNA will go and bind only at the sequence where there is the presence of this PAM sequence, that is the protospacer adjacent motif. So while practically also performing this technique, we have to keep in mind about the PAM sequence, or I will say that this is the one of the crucial thing to determine. We cannot randomly design any guide RNA that okay, there is 1,000 base pair of gene, I will select any 20 base pair. No, we have to see that where is the PAM sequence is there. Okay, so this is the protospacer adjacent motif. It is a short DNA sequence which ranges from 2 to 6 nucleotide that follow the DNA region targeted for cleavage by CRISPR system. In most of the cases, as I already told you, that most of the people using SP-Cas9, that is the Streptococcus pyogenes, the PAM sequence is the NGG. What this N stand for? N stand for any nucleotide from A to G C. Here it may A, it may T, it may G, or it may C. Like it will be AGG, TGG, GGG, or CGG. Okay, the sequence, like I say that I have selected 120 base pair sequence, it will start from 5 prime to 3 prime. Then at the end of 3 prime, there should be present of this NGG. Okay, and I told that N that stand for ATGC. So sp-Cas9, this Cas protein will recognize this NGG sequence. It will come and it will bind over there and it will do the cutting. It will not do any random cutting at any places. Okay. So these are the different PAM sequence of different Cas9. Okay. There are, as I told you, that after sp-Cas9, there is a list of different bacteria in which we have identified different type of CRISPR-Cas system. Okay, so these are the system, and here these are the different type of PAM sequence. Okay, one important is this sp-Cas9, and apart from that, we are also going to discuss about the CPF. So for the CPF, it is a T-rich sequence. Okay, basically, and apart from that, apart from T, that should be any ATGC. So here also you can find the triple TV. It should be 2T. It may be 4T. Okay, what it means that if there are 4T and there is a complementary of 20 base pair guide RNA upstream of them, if they have a homology, then Cas9 will be recruited, CPF will be recruited. It will come and it will identify it and it will cut the DNA, or it will do its activity, whatever it is going to destine. So here again, in another form of representation, this is the single guide RNA. This is the single guide RNA. This is single guide RNA and Cas9. This is our NGG, as I told you that it is a PAM sequence. Third is the target specific cleavage. It has bind over. This is the PAM sequence. This is part of guide RNA. This has a very, or we can say, precise location in the domain of Cas9. It will fit every part of that. It will fit and it will do the cleavage. Okay, and here also, after cleaving this particular activity, as I told you, in most of the cases or 90% of the cases, the pathway of repair that is going to activated inside the cell is the NHEG, non-homologous end joining, and this also thing is also referred as the knockout. Okay, there are two different approaches are there: knockout and knock in. As the name suggests, knockout, that means we are going to disturb the gene function, and knock in, that means we are going to enhance its function by overexpression or by any of the means, or we can say by adding one more uh, sequence that is going to transcribe the protein, etc. Next, again, same thing, the methodology, designing of a guide RNA, and there is a Cas9 nuclease. And if we are going to use the Cas9, if it is sp-Cas9, then the PAM sequence will be NGG. Again, this type of structure is going to form inside the cell in any of the system that you are going to use. See, as I told you that Cas9, it is also a kind of a complicated protein. It has a different domains in that. One domain is so this is the RAC lobe, NUC lobe, recognition lobe, and nucleus. That this is the part of Cas9 that is going to cleave the DNA. And RAC lobe, that means it is going to recognize. What it will recognize? It will only think about the two things, that where there is the guide RNA का ट्रेसर RNA का part कहाँ पर है. One thing is that, and another is where is the PAM sequence. Once it will be identified with the help of this lobe, REC lobe. Then once it will find and it will fit, then this cleavage lobe will be cleave this activity. Okay, and apart from that, there are the HNS domain, that is histidine rich domain, and this is the RUC like domain. So these are the domains or active part of the protein. You must have studied about the structure of protein, primary, secondary, tertiary, etc. And in the tertiary quaternary, in quaternary structure, there are the different type of active or active domains. They will be combined together and they will perform the same activity. So you can say this is a quaternary structure of the protein. These are the different steps that how you can perform inside the lab. Okay, most of the things that I have already told you, but here we can say that we can discuss each and every part little bit in detail. So first of all, even before going to identifying the PAM sequence, what you need to identify is the target gene. Okay, first you should be clear in your mind that which type of which gene you are going to which gene you are going to target. One thing is that, and the second thing is that what you want to do with that gene. You want to knock out that gene, you want to knock in that gene, or you want to suppress that gene. Okay, there are the three things. In case of knockout, I told you that it is completely switching off the gene. In knock in, we can say we can enhance its activity. Okay, and third, that is the lowering its effect, suppression. Like one gene is I want that this gene is going to transcribe and produce one product, but not in that much amount. I want that it should be in a lower range, or प्रोटीन का जो कंसंट्रेशन वो इतना नहीं होना चाहिए, इट शुड बी कंपैरेटिवली लेसर. सो वंस you are clear that you want to do knockout, okay, of one particular target gene. If I say that what the thing that we are going in my lab, that I will say that I will give one example of that only. Like we have I can say that there is a carotenoids are there, right? Okay, carotenoids that in plant, you all know that color of leaf is the green. Okay, so there must be some pathway which are going to produce this green color pigment. That pathway is the carotenoid pathway. In that pathway, one gene is there which is very important for production of this green color. And we also know that if that green color will not produce, chlorophyll will not produce, plant will not able to do photosynthesis, it will die. Okay, so I am going to decide that I want to see that whether this CRISPR system is working in my plant or not. So what I will do, I will target that gene which is responsible or which play a key role in the pathway of production of the carotenoid or production of the green color. I have identified, I have extracted. Now not extracted, I have identified that yes, I want to target the PDS gene. Okay, PDS stand for phytin desaturase, okay, which is one of the key regulatory enzyme in the carotenoid pathway. Okay, so next step, I want the sequence of it. Okay, so in I don't know you people know or आपको पता है कि नहीं, but today after the sequencing method there is a list of advancement in the sequencing also, and we have a list of different plants and animals of which we know the complete genome of it. Okay, so whether it is Arabidopsis, rice, wheat, maize, banana, etc., tomato, there is a list of different plants are there जिसका genome sequence हमें पता है. So if you are working on a crop which sequence is already know, then it is beneficial for you. Okay, so again come back to my example, that is banana. That in banana we have a, we know the complete sequence of it. Okay, and that thing is present in the banana genome hub. If you go on internet and search, then you will find the banana, you will search that banana genome hub, you will find the entire site, and you can see that different locus, and you can try to explore the things. Okay, so once I know that I want to target the PDS gene, what now I will do, I know the sequence of it. Yes, I have a banana genome hub, I will extract it from that. Okay, so directly you cannot do that. What you have to do, you people have studied in the bioinformatics that BLAST is there, basic local alignment search tool. What you have to do, you have to extracted the sequences of gene of various model organisms. You all know that Arabidopsis, tobacco, these are the model organism. Okay, so you have to select 5 to 6 different plants which sequence is already known, whether they are model plant or not. You have to take all those sequences, you have to multi align in the banana genome hub. You will find certain hits out of that. Okay, you will find certain hits out of that, and then you will come to know that yes, this is the 1,000 base pair thing that is going to produce my PDS protein. You got the sequences. Now the next step is identifying the PAM sequence. To identify the PAM sequence, there is a, there are also list of different software bioinformatics tool are there, not software, bioinformatics tools are there. These are the different tools like chop breaking case. There are a lot of them, most of them they are efficient, you can use any of them. Choose any of this software. जो भी आपको 1,000 बेस पेयर का सीक्वेंस आपको मिला था बनाना जीनोम हब से, आप उस क्वेरी को यहाँ पर पुट कीजिए. Then this tool, they will give a hit of that, or it will give a list of different guide RNAs in which you will find that yes, 20 base pair sequence is there, and there is downstream of that there is the PAM sequence. Okay, so might be possible in the 1,000 base pair gene you will find 100 different uh, sgRNA, and out of that you have to select that which you want to, which you are going to utilize. Okay, now after that, once you decided that yes, I am going to use this third number of sgRNA that is listed by breaking cas tool, then what you have to do, you have to cloning the sgRNA into a suitable CRISPR-Cas vector. Okay, if you guys interested, then you can also search on internet about the CRISPR-Cas vector that are used in lab. Then you will find a list of different CRISPR-Cas vectors. Some of them they are specific to plant, some of specific to monocode, some dicode, एनिमल्स के लिए अलग है, मैमलियंस के लिए अलग है. इफ यू वांट टू incorporate this system in bacteria, there is a different set of vectors are there. If you want to do transgene, then there is a different type of vectors are there. So according to that, you have to choose. And in cloning method, the all the different molecular biology technique, that is ligation, restriction, digestion, ligation, cloning, transformation, everything will come, and you will find, or you will design your CRISPR construct vector by cloning method, molecular biology techniques, with the help of molecular biology techniques. After you will find that yes, my CRISPR construct is ready now. Okay, see here, they have given here I have given you two different example. One is the single gene target, another is the multi gene target. Okay, I have already done this single gene target, and I am right now trying to do this multi gene target. What this multi gene target means, not just one single guide RNA, but I am trying to use three different type of sgRNA, which is going to uh, target one gene, two gene, and three gene at the same time. Okay, so this is the multi-gene target strategy. This is the single gene target strategy. Whatever your strategy is, this is the vector. I'm also going to explain this thing in later on slides also. So once you are decided that your CRISPR construct has been formed, you have done single gene targeting, then you have to confirm your vector, and after that you have to do the screening of then you have to introduce this vector into the host species via transformation. Okay, like if I say that in case of crops, you have studied about different system of transformation, like agrobacterium mediated transformation, then biolistic method, particle bombardment, etc. Okay, so you can choose any of the method which is feasible for your crop. Okay, and after introduction of that, you have to grow your plants whichever time it will take. Some plant take some plant get mature in less time, some take years also. So after that, after you will find that plants has been generated, you have to screen those putative edited lines, that yes, I am saying that this might be my positive line, so you have to screen them. And after that screening, you will check the Cas9 gene. If the Cas9 gene is present in your plant, that means that gene has been inserted and my CRISPR system has been introduced, and it, मैं ये होप लेके चलूंगी कि उसने अपना काम किया होगा. And after that, what you have to do, validation of the edited line. And for validation, you can go for sequencing. That is the ultimate way to confirm your plants. So this is the delivery method, as I told you: genetic transformation, biolistic method, floral dip. Floral dip is particularly used in case of Arabidopsis. And apart from that, micro injection, protoplast. This protoplast also I am working on. After that, screening and confirmation of transgenic lines. And finally, if you find out that yes, my there is a type of deletion or addition in my target gene sequence, wahi same sequence that you have taken or you have chosen from the in silico method. Okay, that sequence will be with you. So you have to see that there is a type of differences at target sequence. Then you will see that there is some addition of 2 to 3 base pair, 10 base pair is deleted, something like that. Then you can say that yes, my plants are transformed. And after that, you will go for the further evaluation. Like if I say that I have selected, or I have to see that whether my plant will produce green color or carotene or not. Then in morphologically also, phenotypically also, you will find there is the production of albino plant or plant with the variegated leaves. That means that PDS gene has been disrupted, and it has not worked out, and so there is no production of any carotenoid plants. मेरा albino रह गया, और in future also it is not going, अगर मैं उसको acclimatize भी करती हूँ, then it will not able to survive. So that means CRISPR system has worked and PDS gene has been knocked out. So this is the sync for this. Up till now, I want to show one video of this to clear these things.
Of our genome, over 20,000 genes, 3 billion letters of DNA. DNA consists of two strands twisted into a double helix and held together by a simple pairing rule. A pairs with T and G pairs with C. Our genes shape who we are as individuals and as a species. Genes also have profound effects on health, and thanks to advances in DNA sequencing, researchers have identified thousands of genes that affect our risk of disease. To understand how genes work, researchers need ways to control them. Changing genes in living cells is not easy, but recently a new method has been developed that promises to dramatically improve our ability to edit the DNA of any species, including humans. The CRISPR method is based on a natural system used by bacteria to protect themselves from infection by viruses. When the bacterium detects the presence of virus DNA, it produces two types of short RNA, one of which contains a sequence that matches that of the invading virus. These two RNAs form a complex with a protein called Cas9. Cas9 is a nuclease, a type of enzyme that can cut DNA. When the matching sequence, known as a guide RNA, finds its target within the viral genome, the Cas9 cuts the target DNA, disabling the virus. Over the past few years, researchers studying this system realized that it could be engineered to cut not just viral DNA, but any DNA sequence at a precisely chosen location by changing the guide RNA to match the target. And this can be done not just in a test tube, but also within the nucleus of a living cell. Once inside the nucleus, the resulting complex will lock onto a short sequence known as the PAM. The Cas9 will unzip the DNA and match it to its target RNA. If the match is complete, the Cas9 will use two tiny molecular scissors to cut the DNA. When this happens, the cell tries to repair the cut, but the repair process is error-prone, leading to mutations that can disable the gene, allowing researchers to understand its function. These mutations are random, but sometimes researchers need to be more precise, for example, by replacing a mutant gene with a healthy copy. This can be done by adding another piece of DNA that carries the desired sequence. Once the CRISPR system has made a cut, this DNA template can pair up with the cut ends, recombining and replacing the original sequence with the new version. All this can be done in cultured cells, including stem cells, that can give rise to many different cell types. It can also be done in a fertilized egg, allowing the creation of transgenic animals with targeted mutations. And unlike previous methods, CRISPR can be used to target many genes at once, a big advantage for studying complex human diseases that are caused not by a single mutation, but by many genes acting together. These methods are being improved rapidly and will have many applications in basic research, in drug development, in agriculture, and perhaps eventually for treating human patients with genetic disease.
So I hope that this video clears concept of CRISPR to you, or I'm able to explain these things. Now, next is this is all about how the system works. Next is application of this system in the crop improvement. Now we will see that in which different way this system can be implemented in the plant, or we will also see some of the example worldwide that are that has already done this thing to improve certain trait in the plants, and we will also discuss a little bit about the cpf1. Okay, so this system has been used in plant breeding for various reasons, such as the to insert point mutation, which is similar to the natural SNP. SNP, that means single nucleotide polymorphism. And why it is referred as natural SNP? Because at the starting of lecture, I told you that mutation has been there in the environment before the existence of human, also before the existence of human. So plants were trying to adapt the things, and they were evoluting. Okay, evolution was there before also, and that that thing was occurring in very slower manner, and it was in the form of SNPs. So we can say that even we don't want to chop out a large part of a gene. Instead of that, we can also change one single nucleotide also with the help of this technique. Okay, and what will happen if मतलब एक nucleotide change करने से भी क्या होगा? So for that, if I say that you all have heard about the uh, genetic uh, genetic background, or genetics of the disease called sickle cell anemia. In that, you know that there is only changes in the one nucleotide, and there is the formation of altered amino acid, and in that way the hemoglobin will not form brown, and instead of that it is in the sickle cell form. And we know that if it is in the sickle cell form, it is not going to perform function, and that person, if it is in a severe condition, that person will die. So in certain situation, in certain genetic disorder where it is only due to change in the one nucleotide only, in those cases we can definitely use this technique to cover those things, or we can say to get ready made to overcome those diseases. Okay, so here this part of single mutation will play. Apart from that, to make small modification to gene function, as I already told you, that we can also even suppress the gene, that we will not completely knock out, we will not overexpress it, but we want that it will express, but in a certain or in a slower rate. Then next, for integration of foreign genes, okay, that is a kind of a transgene, we can say, for gene pyramid and knockout, for the repression or activation of gene expression, as well as the epigenetic editing. Okay, epigenetic editing, I hope that you know that what is epigenetic is that epigenetic, that means change in the genome, or change in the DNA sequence due to the environmental effect. That is what epigenetic means. So epigenetic editing we can also perform with this technique. For this, there is example like the use of zinc finger nuclease, which is one of the genetic modification technique apart from CRISPR-Cas. It has been used in Arabidopsis and maize, that has led to successful development of herbicide-tolerant genotype through insertion of herbicide resistance gene into target site in the genome. Okay, you all know that there are a lot of herbicides are there that are used in the agricultural field to to herbicide to remove the herbs especially, and to increase the production of our desired crop. But this herbicide, they have a broad range, so they also affect, or they also kill our target plant. So here, with the help of this technique, we can introduce the gene which is resistance to the herbicide that we are going to introduce in our desired plant, and so that that herbicide will not affect our plant. Next is the using CRISPR-Cas technique. Jiang et al. They have obtained a biotic oil form, that is Camelina sativa seed, with an improved fatty acid composition, which make it more beneficial to the human health. It is more resistant to oxidation and more appropriate for the production of certain commercial chemicals, including biofuels. Next, a major area of application of gene editing approach in plant breeding is to create variety resistance to plant pathogen or pest. This thing I also told you in the beginning, that this technique is used to resist from various pathogen, various abiotic stresses, etc. So for that, this can be achieved by modifying susceptibility gene, resistance gene, gene regulating the interaction between effector and target. Okay, then the gene regulation plant hormonal balance also. For example, wheat genotype resistance to powdery mildew disease were obtained by talent and CRISPR mediated gene editing via mildew resistance locus. Okay, this is the locus which is responsible for this disease. Okay, and we can knock out this locus, and so we can say that now that wheat variety is going to resist with this mildew, which is done by which is the causative agent is the fungus. Genome editing technique have also been used to produce a plant resistance to bacterial leaf blight that is caused by the xanthomonas. So these are the some examples in which this technique has been used to overcome the disease. These are the other certain plants, list of plants that has been modified with the help of CRISPR-Cas technique, and most of them they are the resistance to some mainly disease only. Then, apart from that one, another example is the CRISPR-Cas system has been investigated for its efficacy in providing interference against Gemini virus by using a transient transformation system in the Nicotiana, degradation suppression of curly top genome by single guide RNA that has been demonstrated. This is one more example. So now let's go to the second part. Just a minute. Just a 2 minute. The second part of the slide is uploaded. So meanwhile, I can say that this system has been used worldwide in a very rapid manner, and in our lab also we are doing on the same thing, and we have also succeeded at in some of the up to the some level for increased production of beta carotene in banana. Here our main aim is not to uh, is not like any disease resistance or abiotic stresses, but main is the improvement of nutrient content. Okay, so here our main aim is to increase beta carotene, so that any people who eat that enrich beta carotene rich banana, then it will help them to get the vitamin A, especially because blindness is one of the major disease in the India due to malnutrition. So this is our very small effort to overcome the malnutrition. And well, basically in the second step we are just going to discuss more about the other applications especially, and a little bit of my work that I am performing right now. Just a minute, the file PPT is uploading.
Hiral, just wait for a few seconds. The size of PPT is too big, so it takes a time.
Okay, no problem.
So
It is
meanwhile, you
actually
can share
too.
your experience how your institute and that all. So how
Sure.
is the work going on, and on which
Well, the
topic that
work
you are doing a
is
research?
the work is going a little bit in the pace, I can say. But yes, definitely we have to work hard on the daily basis, like every day reading new research paper, interacting with others, so that how we can improve. And we can also say that, like attending any conferences and symposiums, so that we can get updated that what is going at global level. So yes, these are the things that we are doing in routine, I can say. And definitely trial and error for different experiment, it is ongoing. And in 99% cases, initially we get a failure only. So only thing that we have to be is optimistic. Okay, our slide is uploaded, so let's start the second part. Please share it.
And sorry for the trouble, Hiral.
No problem. Okay, let's start. As I told you that here we are just going to discuss about the main applications. Mainly three applications: gene disruption, insertion, and regulation. We will see little bit of each and everything in detail about them. First is the disruption. As I already told you, that disruption, that means we are going to knock out, that we don't want to activate this gene, or we don't want any product of this gene. And moreover, I can say that there is some hypothetical gene that this is might be going to do this type of activity, but that is just my assumption. So if I want to make sure that yes, my hypothesis is correct, then what I will do, I will go with this technique. So I will completely knock out this thing, and whatever the expected outcome was there, if it is not there, then definitely जो मैंने सोचा था कि ये जीन hypothetically ये function करता है, that means the function validation will be there. And in most of the cases, this technique is also used for the functional validation of any gene, important gene, so that in future we can do any other things with that gene. This technique is also powerful as it can inactivate any potential harmful or non-beneficial gene that downgrade the quality of a plant. Like if I say that in maize, the corn of maize, I find that there are lot of scales are there on them. Then if I come to know that which gene is responsible for the production of more number of that scales over that, then I will disrupt that gene, so that now there will be no any extra scale will be there. So this type of, if I find that this trait is not so good, or I don't want this trait to be in that, so we can definitely disrupt that genes. Gene disruption is most applied technique, and it can knock out genes simply by small deletion or insertion via the NHEG. Okay. Next, see, this is the standard example of it. Like as you can see over here, that these are the it has a perfect waxy structure of cone, right? So the maize, the for this waxy appearance of maize, it is due to the WX1 gene that encode a starch synthesizing protein that involve in the kernel maintenance. Kernel maintenance, that means each and every kernel of this code. Okay, but today there is a known mutant maize that has a deletion in the coding sequence of this WX1 gene that cause it to have an altered grain starch composition. Okay, and this, because of this altered grain starch composition, there is a appearance of matlab, each and every kernel of corn, it will look very similar, it will look attractive, and it has a slight different composition of starch. You all know that starch has a two main chains, amylose and amylopectin. Then with the help of like proportion of this amylose and amylose pectin is different in case of the normal or wild variety of corn, but with the, or we can say by disruption of this gene, there is a alteration in proportion of amylose and amylopectin of the starch, and this thing will make appearance of this kernel in this type of waxy structure. Like here, as you can see that this is the WX gene, and what this WX gene is responsible, it is going to produce the amylose. But as this red color arrow, it shows that we are going to disrupt this gene function, and because of that, the normal yellow dent, that is the single kernel of maize, it appears like this because it has 75% amylopectin and 25% amylose. But by deletion of this, or sorry, not deletion, but disruption of this gene, it will make 100% amylopectin, and it will give this translucent appearance of the kernel. So this is also in uh, a very recent one. You guys can also search on internet about this vexicon. You will come to know about many different aspect of this. So, attempt to introduce this mutant LL by crossbreeding a non-allied vexicon with an allied plant with excellent agronomic quality, it were unsuccessful. Like this type of thing has already been attempted via breeding, but they were unsuccessful. But DuPont company, they induces gene disruption by CRISPR-Cas, and they it work in that. And how it work? It by deleting the entire WX1 gene with the usage of two Cas9 guide RNA. As I told you that you can also design one guide RNA. You can also design two guide RNA, three. You can do any of them. Like in paper, they have also used up to 62 guide RNA at the same time. So you can imagine that how different things you can do with this technique. Each of the Cas9 guide RNA, it will target two sites, and which are the upstream of transcriptional start site and the downstream of the stop codon. And then the region is excised, and the remaining DNA damage is repaired through the NHEJ, and which will bring about this WX1 null allele. Null allele, that means this allele is present but is not functioning with the vaccine phenotype. This is like how they have used in a tradition way, and how this CRISPR has worked, like by doing many breeding attempt day by day, and it also take years, but still also they were not able to got not able to get those type of phenotypic appearance. But with the help of CRISPR, they did it in a very few time period. So this is the advantage. Okay, this slide is of the work of from my lab itself. So I will just summarize these things, and I already gave you example of this. I told you about the PDS gene. So this is the PDS, this is the single guide RNA of that PDS gene that is going to produce the green color. This is the control plant. Okay, what we have done, we have constructed, this is the CRISPR construct vector. We have constructed or ligated a target DNA or target guide RNA in the CRISPR construct, and we have agrobacterium transform in the banana ECS. ECS stand for embryogenic cell suspension, and banana take 7 to 8 months to get the mature plants out of the ECS. And in the final stages, we found out that most of the plant, they were white in color, not white, but if I use the scientific term, that they are in the albino in color. Some of them, they are total albino. Some were having a variegated leaf. Variegated, that means patches of green and white color. So what this shows that PDS gene has been disrupted by CRISPR, and now it is not going to function, and it will not function, and there will be no any production of green color. And these things we have identified by the sequencing. Like this type of plant, we got many plants, and we have done the sequencing of it, and we found out the various insertion and deletion in all these lines. So that means that gene has been damaged and it is not functioning. So we can say by this, our main function was to validate that whether CRISPR system work in the banana or not with the help of phenotypic changes. And after successful introduction of CRISPR in the banana plant, we have targeted another gene which enhance the beta carotene. And recently also we have also targeted one another gene. This is the again one more work from our lab, and in this work has also major part has been also done by me, and this has been recently published in the Journal of Experimental Botany. In this we have also isolated protoplast. We have constructed CRISPR, but here the gene that we have targeted, it was ccd4 carotenoid cleavage dioxygenase. And ultimate goal was also to increase the beta carotene and to reveal the function of ccd gene. That yes, definitely, that like it was hypothesized that ccd is going to work on beta carotene. Okay, so what we have done, we have knocked out this gene, and we found out that expression of this gene was decreased, and so beta carotene level was increased. So that means ccd is going to degrade the beta carotene. So here we have done two things: unravel the function of ccd4, and at the same time increase the beta carotene in the banana. So initially here also we have done in the two different way. One we have done in the protoplast also, but protoplast regeneration is challenging, so we have used the particle bombardment method, biolystic method, for the transformation of banana. First was the gene disruption. Second is gene insertion. Gene insertion, that is the addition of or addition, is another famous technique where more than one nucleotide base pair are added into a DNA sequence. And as I already said, that here whatever the DNA sequence that we are going to introduce, it will be of our choice. Okay, so this is how it will work. These are the certain example of it, that like in TC1507, this is the name of variety of maize that contain both bt gene that confirm insect resistance, and second is the acetyl transferase, that is herbicide tolerance. Next, to date, the most common and efficient method used is to co-locate all the desired gene. Co-locate all the desired gene. That means, like, I know that there is a list of different genes which are going to play a very important role for one function. So what I will do, I will collect all of that genes, and I will try to align side by side. That is also referred as the gene stacking. So the co-location, obviously, it's not an easy task. So each of the gene may potentially affect each other if they are placed. Next is the gene regulation. And in gene regulation, it is a technique where the gene is encoding for a transcriptional factor is altered to induce the changes in its expression level. And here we can say that plant traits such as fruit color, size, shape, kernel size, etc., that can also be regulated. Next is the CRISPR system can also be used to regulate expression of gene in the plant. Here, the another part of our today's discussion, that is Cas9 and cpf1. Here, the role of cpf will come. While studying on the CRISPR-Cas9, scientists also found out that what once see, this is the adaptive immune system that is adapted by the bacteria. But what virus have also done? Virus have developed anti-CRISPR system. Okay, like it is like a war between bacteria and viruses. So what viruses have done, they have developed anti-CRISPR system, and in anti-CRISPR system what they are doing, they are mutating the cas9 protein, and it is that cas9 is referred as the dead cas9, dcas9. What this cas9 is there, it will bind, everything will be same, but there is no any capacity to do the cutting. So here you can say that everything will be there, but there will be no any cutting or chopping of the DNA. So what we have done, we have utilized this system to, we have, like we can say that we have also uh, manipulate this system in a like हमने इसको ऐसे modified किया कि we can also use this function for our purpose. Here what we will do, dcas9 के साथ में हम लोग कोई भी अलग से system जोड़ देंगे which may be effector or activator or suppressor of another gene. So what will happen, the things will be there, but that effector will work and that gene will be overexpressed or underexpressed. So this is the schematic representation of that. Next is the base substitution. This is also one of the very recent thing I can say. To summarize this thing, base addition, base addition and CPF1, it has recently introduced in the 2020 and 2021. In base addition, it is of two types: CP, that is cytidine base addition, and adenine base addition. Here, what we will do, this nicase Cas9 and or death dcas9, इसके साथ में हम लोग क्या करेंगे, we will also incorporate of our enzyme CB or AD, and dcas9, it will just over there, it will not cut the things, but our deaminase enzyme CBE or AB, what they will do, C की जगह पर they will introduce U, एंड सी की जगह पर यू इंट्रोड्यूस हो जाएगा, और जब ये चीज नेक्स्ट लेवल पे जब डीएनए रेप्लिकेशन होगा, देन इट विल बी टी. So we can say that there is the C to T substitution, and in adenine base emission, we can say that there will be substitution of A to G. Okay, this thing is highly recommended right now. Why? Because there is only changes in a one single nucleotide, and this thing can be utilized in cure to cure the various genetic disorder which are only due to single base pair mutations. This is a kind of detailed description of it. Next is the CRISPR CPF. This CRISPR CPF, it stand for the Croptel and Francilla. Okay, these are the two different like bacteria. It is like combination of it. Okay, if I go precise and summarize this thing, then CRISPR CPF, there is a little bit difference in between SPCas9, CRISPR-Cas9 and CPF1. What is the main difference is, one is the PAM sequence. I hope you are clear with what is PAM sequence, proto-adjacent motif. In case of Cas9, it is G-rich, that is NGG. But in case of CPF, there is the high T-rich PAM sequence is there. Okay, so this is the major difference. And apart from that, another difference is you can see that the length of guide RNA. Here you will see the length of guide RNA, and here you can see the length of guide RNA. In case of CRISPR CPF, only CRISPR RNA is there, no tracer RNA is there. But here there is the combination of CRISPR and tracer RNA. So we can say that to reduce the size of DNA in the construction of vector, this is highly beneficial. So first advantage is the T-rich PAM sequence. Second is the smaller length of guide RNA, and third and most important thing is the production of the sticky end. In case of CRISPR-Cas, there is always blunt, maximum, not always, but in 99% cases there is the blunt end and very less sticky end. So initially I said that in most of the thing, एक बार CRISPR से जीनोम में एडिटिंग हो गई, then there is only activation of random insertion. कभी भी homology directed repair नहीं होता है. The reason behind that is the production of blunt end. But if I want to introduce a DNA sequence of my choice in plant, then I will go for CRISPR CPF. Why? Because after cutting of this after cutting with the CRISPR CPF, there will be generation of sticky end. So I know that what type of sticky ends that are going to generate. So इसके साथ में what I will do, I will synthesize, or I will produce a donor template DNA that will be complementary to this. So there will be now जो नया जीन जो नया रेप्लिकेशन इसके बाद में होगा इस डीएनए का, देयर विल बी फॉर्मेशन ऑफ सीक्वेंस इन विच माय edit or my desired gene or my desired sequence will be there, so it will going to function in the way that I want. So this is the best thing about the CPF, and nowadays people are also moving from CRISPR-Cas9 to CPF1 only because of this reason. So this recently there are again two different version of CPF are there, that is LB-CPF and AS-CPF. These are the two variant of two different bacterium. They have their own advantages and disadvantages, and recently this CPF, different type of CPS has been introduced in different plant, like LB-CPF, it has generated mutation in rice, and this AS-CPF, it has been used in rice as well as soybean. So again, this is the same thing that when tested for their ability to introduce target gene via homology directed repair, this CPF nuclease generate precise gene insertion at the target site so that we can incorporate our desired donor template. And recently, to expand the scope of this CPF1 mediated genome editing in rice, this Lee et al. Scientists, they have developed LB-CPF-RR variant that enable the editing and multiplex editing of target gene containing TYCV PAM, that is TYCV PAM, that is the T-rich sequence, which is going to uh, uh, identify by CPF-Cas9 protein. So, like the CRISPR system, CPF1 system, it may be combined with the base editing and DNA-free genome editing. Like CPF के साथ में हम लोग here cas protein को ना use करके हम लोग D-cas और N-cas को use कर सकते हैं. And apart from that, CRISPR CPF1 mediated base editing, they use the T-rich sequence, so that T-rich sequence is comparatively higher in some of the gene sequence, so we have more options to choose the guide RNA. And so it has, we can T-rich PAM sequence, it has produced C to T conversion in human cell. Similar application in crop plant should not be too far in the future. And these are the some examples of crop that are recently we can say done or recently improved by this of these are the respective organization of them, and some of them they are introduced in the market, and some of them they are under the pipeline, and some of them they are might be in very near future. You will find that this genome edited plants are in the market, and we are using them. So might be possible after 10 year, if you go in market and if you see that there are vaxicones are there, then you must not be surprised, because this thing is the future. So thank you all of you for your cooperation and listening me quietly, peacefully.
Thank you, Hiral, for the wonderful session. And I must say that you are passionate about your subject
Thank you.
and you explained very well. So now it's time for the question answer session. There
Okay.
are two questions. That one is from Jill Modi, and second question is from Charmi. So let's take one by one. So
Okay.
first question is it possible to insert a selection marker or fluorescent marker into a pre-existing Cas9 CRISPR plasmid?
Yes, definitely you can do. You can do any type of editing. See, hypothetically, you can do any of the things, but for that you need a very like a hard work. You have to go and search on literature. But yes, definitely this thing is possible. If you have already CRISPR-Cas9 vector, then you can definitely incorporate your inflorescence protein. And I will suggest that yes, definitely already paper publish है इसके ऊपर बहुत सारे, आप उसको देख सकते हो और आप उसको उस चीज को incorporate भी कर सकते हो.
Now, come to the second question. Are CRISPR technology or other such enzymes going to replace transgenic technology in plants and GMO in coming years? So, is it?
डेफिनेटली इफ यू हैव लिसन माय कन्वर्जेशन, मतलब एंड तक अगर आपने सुना होगा, तो अभी जस्ट रिसेंटली मैंने ये चीज़ आपको बताई दैट यस डेफिनेटली दिस टेक्नोलॉजी एंड नॉट अदर सच एंजाइम्स, लाइक इट्स नॉट अदर सच एंजाइम, इट इज एवरीथिंग विल बी कम अंडर दिस CRISPR only. Okay, and definitely they are going to replace the transgenic, and it is replacing transgenic technology in plants and GMO in the coming years. And if I say that in Japan and USA already उसमें उन लोगों ने बहुत सारी क्रॉप को already approve कर दिया है, and they are also using or they are eating these crops, but they are not genetically modified, they are genome edited, so there is a vast difference in between them.
So, due to shortage of the time, okay, we are moving towards the ending of the session. So, one student, okay, he wants to ask a question directly. So, let me take
Okay.
on the screen. अब आ जाइए ध्रुवित, फटाफट नहीं अभी जा. Best chair पर.
Ma'am, would you kindly? Ma'am,
You're audible, you can ask.
Would you kindly determine the role of CRISPR in vaccines And would you kindly shed some light on role of CRISPR and its application regarding coronavirus, COVID-19?
Okay, yes, it see the coronavirus. It has we know the variants of. We know that there are a lot of variants of coronaviruses are there already, okay, and day by day new and new variants are coming. So in most of that, first what we have to do, that we have to do the in silico study, and in our lab we have also done the in silico study of the coronaviruses, and that paper is also going to publish in the near future. And we are also trying to prepare a plant based vaccine, but in this case here CRISPR role is not in a direct way. CRISPR role is not that in a direct way for production of this edible vaccine, but definitely we can utilize this CRISPR system for production of vaccine, but it is quite complicated also, but definitely we can do also.
Ma'am,
Yes.
regarding CRISPR's role in DNA vaccines, US companies they have specifically manufactured DNA vaccines and are also in consumption in United States. So, ma'am, can you shed some light on that, because that seems to be the new area for CRISPR's utilization?
See, definitely. I can say, see, CRISPR-Cas role, it cannot directly implemented for the production of vaccine. Okay, but yes, which part of virus is going to produce that epitope? किसके अगेंस्ट में हमें एंटीबॉडी चाहिए? कितना पार्ट हमें डीएनए वैक्सीन के लिए बनाना चाहिए? उन सारी स्टडी, उस सारे एपिटोप्स को आइडेंटिफाई करने के लिए क्रिस्पर का रोल 100% है, और बहुत सारी जगहों पर बहुत सारी लैब के अंदर इसका रोल यूज भी हो रहा है. उसमें भी सेम चीज होगी कि पूरा जो हमारा कोरोना वायरसेस है उसके जितने भी हमने आइडेंटिफाई किए हैं कि भाई दिस आर दी एपिटोप्स दैट माइट बी पॉसिबल कि जो हमारे बॉडी के अंदर एंटर होते हैं और जो भी सारी इम्यूनिटी और मतलब हमारी इम्यूनिटी को एक्टिवेट करते हैं, तो उसके अगेंस्ट में उस पार्ट को आइडेंटिफाई करने के लिए डेफिनेटली हम लोग क्रिस्पर को यूज कर सकते हैं. तो यहाँ पर क्रिस्पर का बहुत बड़ा रोल होता है. सो इन दैट वे डेफिनेटली डायरेक्टली और इनडायरेक्टली क्रिस्पर इज देयर. एंड देर इज मतलब बहुत सारे पेपर्स भी, अगर आप यू विल फाइंड अ लॉट ऑफ पेपर ऑन दिस फॉर वैक्सीन ऑल्सो विच इज प्लांट बेस्ड वैक्सीन. एंड इन फैक्ट इन आर लैब ऑल्सो वी आर ऑल्सो ट्राइंग कि प्लांट बेस्ड वैक्सीन वो बनाने के लिए, बट इट इज ऑल्सो अंडर द पाइपलाइन. इन सिलिको एनालिसिस वी हैव डन पर्टिकुलरली फॉर द इंडियन वेरिएंट्स, जितने भी इंडियन वेरिएंट्स है कोरोना वायरसेस के, उसके रिगार्डिंग. And this can be a definitely new era. And let me tell you one more thing to all of you, that see today OTT platform, you all are aware of different OTT platforms. So Netflix के अंदर if you want, if अगर आप सच में इस डायरेक्शन में कुछ करना चाहते हैं, रिसर्च के डायरेक्शन में कुछ करना चाहते हैं बायोटेक फील्ड में, तो एक सीरीज है अननेचुरल सिलेक्शन करके नेटफ्लिक्स पे. तो यू कैन वॉच, देर आर 4 एपिसोड्स ऑफ दैट. इन दैट यू विल कम टू नो ईच एंड एवरीथिंग. उन्होंने बहुत अच्छे से एक डॉक्यूमेंट्री बनाई है 4 एपिसोड के अंदर. एडवांटेज क्या हो सकते हैं, डिसएडवांटेज क्या हो सकते हैं, फ्यूचर में लोग उसको कैसे मिसयूज कर सकते हैं, कैसे ये सारी चीज़ें बनती है. तब आप आज जो मैंने पूरा लेक्चर दिया उसमें से बहुत सारी चीज़ें कोरिलेट भी कर सकते हैं. सो द नेम ऑफ दैट सीरीज इज अननेचुरल सिलेक्शन. इट इज टोटली एंटायरली बेस्ड ऑन क्रिस्पर कैस सिस्टम. आई होप I clarify your point.
Yes, ma'am.
Thank you, Viral, for accepting our invitation. And we need to thank our coordinator, sir, that he gave us opportunity to conduct a webinar. So one student that she is like to give a vote of thanks. So let her introduce. Yes, Salim. You are perfectly audible. Sorry for the technical issue. That student is here and she will connect from the here only.
Okay.
Good afternoon to everyone. On the behalf of our class and Department of Biotechnology, it is my privilege to it is my privilege to propose a vote of thanks on this seminar. We had an extremely useful day with Hiral ma'am, and thank you all of you for clearing and sharing knowledge with us. And especially thank to Mansi ma'am and Gaurav sir to arrange this useful and very knowledgeable seminar. And thank you so much, all of you, to give a very precious thing. This is time to this time you give to us. So thank you, all of you. And once again, thank you, all of you who. Thank you, ma'am.
Okay, thank you so much. Thank you, Gaurav sir. Thank you,
Thank you
Mansi,
very much.
for inviting me and
And one more
considering
thing,
me
Hiral.
to give a talk. Yes. Yes.
One more thing, Hiral. Few students they are here presenting offline, so they want to say thank you to you. So just give me 1 minute.
Okay. Hello. You all are welcome. I hope that many of you go in your desired field and you can achieve great things in research or any other perspective of biotech with just your passion and hard work.
Okay, fine. Thank you. We are ending our webinar here. Thank you. Bye bye.
Okay, thank you. Bye.